In this video, we're going to talk about what happens between the time that the gametes get released from the male and the female to the time that gastrulation occurs. So let's look at what happens to the gametes first. We'll talk about the ovulated egg. So keep in mind that once the egg cell is ovulated, it's considered a secondary oocyte, and it has not completed that second round of meiosis just yet. Remember, it will have to be fertilized by a sperm cell in order for that second round of meiosis to occur. So as this ovulated egg is traveling down the uterine tube, remember it's having to be carried by cilia that are lining the inside of that uterine tube. That egg gets carried towards the uterus, and it may or may not be fertilized by a sperm cell during this time. Now for sperm cells, they have to do a little bit more work. They're going to have to physically swim or move themselves towards that secondary oocyte. And keep in mind that when ejaculation occurs, this will emit between 200 to 300 million sperm into the vagina, but only about 200 to 300 at the most will ever make it to that secondary oocyte into the uterine, in the uterine tube. So it'll take about 30 minutes for the sperm cells to make it to that uterine tube, and again, most of them will either die or they just won't make it throughout that journey. Again, only about 200 or 300 at the most sperm cells will be able to make it into the uterine tube and potentially fertilize that. Only one is going to fertilize the secondary oocyte, but could potentially make it to that secondary oocyte. Now, before a sperm can actually get to a secondary oocyte and fertilize it, it has to go through a 10-hour process called capacitation. And this is where the membrane of the sperm head has to become weak in order for the nucleus to be able to be released from that sperm cell once it encounters that egg cell, so that the DNA can combine and end up creating a zygote. So sperm cells are relatively stable along their journey. They can survive for up to 6 days in the uterine tube. So they can basically just stick to the mucosal lining of that uterine tube and wait for an egg to be released through ovulation. Now, ideally, you would want to have sexual intercourse between 2 to 3 days of ovulation. That would give you the best chance for pregnancy to occur. But in an ideal world, sperm can survive for up to 6 days in that uterine tube. Now, once a sperm cell encounters the egg cell, or the secondary oocyte, we know that that egg cell is going to be covered by some follicular cells, and so the sperm will have to burrow through these cells in order to get to the zona pellucida. And so this is a layer of gel-like material that surrounds the egg, and once it comes into contact with that, there are some proteins in the zona pellucida that will stimulate the sperm to attach to that membrane and then undergo what's called an acrosome reaction. So this is where the sperm cell will release some gel-dissolving enzymes from the acrosome, and that will help dissolve that gel membrane around the egg to allow the head of the sperm to be able to enter into that secondary oocyte. So once the sperm cell reaches the surface of the egg, then those membranes of the two cells can fuse. The head and part of the tail of the sperm can enter the egg and release the nucleus into the egg cytoplasm. And at that point, this is when fertilization is actually occurring, and the DNA of those two cells can merge together to create that zygote. Now, once one sperm cell enters the egg cell, the egg will prevent any other sperm cells from being able to enter to avoid having too much DNA in that developing zygote. So if fertilization by more than one sperm happened, this would be fatal to the egg, and it would not be able to develop into a fetus. Now, once fertilization occurs, the egg will complete that second round of meiosis. It will shed its excess DNA into that second polar body, and at that point, the nuclei of the sperm and the egg can essentially merge, and their chromosomes can merge into a single diploid set of 23 pairs of chromosomes. And so this is when true fertilization is going to occur. This is when we have the zygote, which is ready to develop into an embryo. So everything that comes from the zygote is going to be called a conceptus, This is going to include the embryo, which is kind of the earliest version, the fetus. The placenta will also come from this collection of cells, the umbilical cord, and all of the membranes associated with that. So here you can kind of see the steps of fertilization. Here's our sperm cell. Again, it has that acrosome that allows it to be able to digest these follicular cells or penetrate through these follicular cells and then digest that gel membrane around the surface of the secondary oocyte. So you can see that sperm cell penetrating those follicular cells, breaking down that gel membrane through that acrosome reaction. The head of the sperm is now fusing with the membrane of the secondary oocyte, and that DNA is being released into the secondary oocyte for it to fuse and end up forming that zygote. And you can see that once this happens, that cortical reaction will take place in the egg cell, and that will prevent any other sperm cells from being able to come into that egg cell. So once fertilization has occurred, we have 3 major stages of development within the fetus. We have what's called the first trimester. This is the first 3-month interval in pregnancy. So happens from fertilization to about week 12 of pregnancy. And during this time, this is when the conceptus is going to be the most vulnerable and potentially exposed to damage. This is when most miscarriages will take place, is during the first trimester. More than half of all embryos actually die during this trimester, and that's, again, because the conceptus is so vulnerable to any kind of stress, any kind of medications, illness in the mother, nutritional deficiencies, anything like that. The embryo is going to be very sensitive to that. Also during this time, this is when a lot of chromosomal defects might be taking place during fertilization, and most of the miscarriages that take place during the first trimester are going to be due to chromosomal defects in that embryo. Now the second trimester is going to take place during weeks 13 through 24. This is when most of the organs and organ systems will complete their development. And this is when you're going to be able to tell whether the fetus is male or female. This is when the fetus is going to begin to look more human, and it's going to have a much better chance of survival by the end of the second trimester. Now the third trimester occurs from week 25 gestation to birth, and this is when the fetus is going to be continuing to grow, to develop. This is when the organs will start to really mature and become fully functional during this third trimester. And at this point, around week 35 is when most fetuses are going to be considered relatively viable. You could have them and probably not have too many health problems in a fetus that's born by 35 weeks. Most of the time you want to try to wait until at least 38 weeks, if at all possible. Week 40 is typically, or is generally considered a full-term baby, but most of the time now you can have the baby between 37 and 38 weeks without any kind of problems, without any kind of breathing problems or anything like that. So let's look at pre-embryonic development. We know there's really no universally agreed definition for the word embryo. We know that this typically happens when the conceptus has 3 primary germ layers, which is around day 16 of development. And we'll talk more about that in just a few slides. But this will essentially extend through about week 8 of gestation. And this is when those 3 layers, those 3 primary germ layers, have formed all of the principal or primary organs of the fetus. Okay, so all the major organs essentially will be developed by week 8 of gestation. They won't be fully formed by then, but they should all be present within the embryo by week 8. And so all development prior to this stage is called the pre-embryonic stage. And this is going to include what we call cleavage, implantation, and embryogenesis. So let's talk about those steps. So in cleavage, this is going to happen right after fertilization as that conceptus is traveling down the uterine tube. It takes about 3 to 4 days for that fertilized conceptus to reach that uterus. And so during that journey, or during that time, that zygote is going to be dividing through what's called cleavage. So this is a series of mitotic divisions that will help divide that single-celled zygote into a ball of smaller and smaller cells. And by the time that conceptus reaches the uterus, it will consist of anywhere between 16 to 32 cells, and it will now be called a morula. So it's not going to be any bigger than a zygote. It's still going to have that fertilization membrane around it, but it's going to consist of more than one cell. It's going to consist of between, again, 16 to 32 cells. Now that morula, once it reaches the uterus, will continue to divide for about 2 or 3 more days until it forms what's called a blastocyst. And so this is going to consist of around 100 cells, which are arranged in a hollow, fluid-filled ball. Now, in that hollow ball, we have what's called an inner cell mass. This is what will end up becoming the embryo later on, and will develop into those 3 primary germ layers. The outer layer of cells that forms that hollow ball is called the trophoblast. This will end up developing into the placenta, which we know will provide nutrients and nourishment to the developing embryo. And so the fertilization membrane will essentially break down, and then the blastocyst will emerge or hatch from that. So here you can see the migration of the egg and the conceptus. So here we have that secondary oocyte that has been ovulated from the ovary, released into this uterine tube. At this point, you can see fertilization occurring, where the DNA is merging and we're forming that single-celled zygote. And then that fertilized egg cell, that zygote, starts going through rounds of cleavage, rapid rounds of mitosis, to end up forming this 16 to 32 cell morula by the time it reaches the uterus. So again, this cleavage process takes about 3 to 4 days roughly. And then we have the morula here present in the uterus, which will continue to go through rounds of mitosis. This outer membrane will start to kind of break down. And you can see that happening here in this early blastocyst. And by the time that fully formed blastocyst has formed, at this point, this is when implantation will occur, and it will implant into that uterine lining of the uterus, that endometrial lining of the uterus. So here in this blastocyst, you can see the inner cell mass in this orange color here. So again, this is the cell mass that will end up developing into those 3 primary germ layers, which will end up developing into the fetus. And then the trophoblast in purple around the edges here, you can see, will end up merging with tissue of the endometrial lining and forming the placenta. Now, twins can sometimes occur during this process. We can have 2 different types of twins: monozygotic twins, which are identical twins, or dizygotic twins, which are non-identical. Now, monozygotic twins happen whenever the embryoblast, which is a fertilized embryo, zygote, it splits into 2 cell masses. So what you have is you have 2 developing embryos that are genetically identical to one another and that are sharing the same placenta. So they're going to look exactly the same. They're going to have the exact same DNA, and as far as monozygotic twins will go. Dizygotic twins are a little bit different. This is when you have 2 egg cells that get released during ovulation in the female's body, and they are both fertilized by separate sperm cells. So you essentially have 2 completely different babies developing at the exact same time, the exact same timing method or process in the female. And so dizygotic twins would not be any more alike than siblings, any other type of sibling would be. They're going to be very different from one another because they have different DNA compositions from one another. And they're each going to have their own placenta as well and develop their own amniotic sacs that they will mature in. So here you can see implantation occurring in this uterine lining. You can see rootlets of this trophoblast here kind of embedding in the uterine lining of the uterus. You can see the trophoblast, and then you can see this inner cell mass, which again will end up developing into those 3 primary germ layers. And so here you can see that inner cell mass. It has gone through more and more rounds of division and has started to develop into those 3 primary germ layers, which consist of the endoderm, colored in yellow here, the mesoderm, colored in red in the middle, and then the ectoderm, colored in blue here. So you can see those 3 primary germ layers are starting to develop from that inner cell mass that originated from implantation. So implantation is going to occur roughly 6 days after fertilization. Again, this is when that blastocyst embeds into the endometrium. This is when the trophoblast cells start growing into that uterine lining, and the endometrium itself will grow over that blastocyst to allow pregnancy to truly begin. Now, once implantation has occurred, at this point, the trophoblast will start to secrete human chorionic gonadotropin, or HCG, which is essentially the pregnancy hormone. This is what we test for whenever we do a pregnancy test to see if that hormone is present in the urine or the blood. Now, the levels of this hormone are going to be relatively low whenever implantation is occurring. They're not going to be high enough really to detect, but those levels will increase day by day, and you will start to be able to see them or detect them in blood or urine as the pregnancy continues on. Also, we know that once this human chorionic gonadotropin starts being produced, this is going to stimulate the corpus luteum, which is in the ovary, to secrete both estrogen and progesterone. Progesterone will prevent the female's body from entering menses, prevent her from going through the menstrual cycle, and this will help maintain that pregnancy early on. Later on, a membrane called the chorion will kind of take over the production of HCG, and at that point the ovaries will become dormant. They will not need to produce any more hormones throughout the remainder of the pregnancy. Now, ectopic pregnancies can occur, and they can be very dangerous for females. This is when the blastocyst implants somewhere other than the uterus. This could happen in the uterine tubes, where implantation occurs there due to some kind of blockage. And that can be very dangerous because if the embryo continues to develop in that uterine tube, it can end up causing the tube to rupture, which can cause severe hemorrhaging and could result even in death in the female if it's not caught early enough. This could also happen in the abdomen. That ovulated cell, could enter into the abdominal cavity rather than into the uterine tube. It could be fertilized in the abdominal cavity and can implant there instead. And so this is pretty rare for this to happen, or for the fetus to even potentially develop outside of the uterus, but it has happened before. Now, gastrulation will occur during implantation. This is when that pre-embryo is still going through rounds of cell division. Those cells are starting to kind of migrate and arrange themselves into 3 primary germ layers, which I showed you earlier. You have that ectoderm, mesoderm, and endoderm. Now, that inner cell mass will develop into what's called an embryonic disc, which is going to consist of 2 cell layers with a nice groove running right down the middle. And so the cells that migrate down into that groove end up forming what's called the endoderm. The upper surface of the structure will become what's called the ectoderm, and then the mesoderm is going to be kind of sandwiched in between those first 2 layers. Now, the mesoderm will end up becoming a very gelatinous-like connective tissue called the mesenchyme, which will develop into many, many structures, many connective tissue structures in the body. We also know that the amnion and the umbilical vesicle will also arise from this layer as well, and at this point, this is when the individual will now be considered an embryo. And so here you can see those layers again. You have the endoderm in this yellow layer here. You have the mesoderm consisting of these red colored cells, and then you have the ectoderm here in blue. So those 3 primary germ layers that develop through gastrulation.